So, before getting into singlet oxygen chemistry, we will see one small rearrangement. Then once we finish that, then we will get into singlet oxygen chemistry. Okay. So, that is your photo freeze rearrangement. This you have studied, right? Freeze rearrangement you know for sure. Normally, if you take freeze rearrangement, what happens? Any idea? Okay, I will just write the molecule so that you can recollect it. What happens? You just take this and if you put aluminum chloride, right? You have studied, right? Yeah, aluminum chloride. So you will see your acyl moving towards your ortho and para, right? That you in that case you will generate what? You get what? Carbocation, right? That is very important. You will generate an acyl carbocation. That is your regular freeze rearrangement. But in photo freeze rearrangement, it's other way around. So if you do the photolysis, same way you end up your ortho and you will end up with your para, similar to your freeze rearrangement, but you get one more product which tells something. You get a type of phenol which you do not see in your freeze rearrangement. You get these three products. So, any idea what it suggests? Yes, decarbonization happens. Yeah, it does not happen like that. Intermolecular, intermolecular, no. So, only thing is that this whole chemistry goes in radical. Okay. So, how it works is that if you take your and if you photolyze this, so in regular one you get an acyl carbocation and freeze rearrangement, but in this case you will get an free radical chemistry. Okay. Now, this is in solvent case. Okay. So, that is more important. This is you call as solvent case. That is why always I talk about diffusion, and these are very important when you talk about diffusion. Let us see, I will just write this molecule here for one more time. These two molecules, okay, these two radicals can diffuse. Okay, diffusion is faster. Before diffusion, if they recombine, okay, then you can see some sort of rearrangement happening. Okay, it does not need that it has to recombine there. It recombines here. It can also recombine your para portion. Okay, but if it escapes, then what happens? You get your phenoxy radical, which can abstract hydrogen from the solvent to give you phenol. Fine. That is how you get. If it can have here, if they recombine, if it is escapes from, if it is escape from the solvent, escape from the cage, then you will get this type of radical. Okay. This then can give you Only thing is you have to remember is that this whole chemistry goes by radical, that is all. Previous case it was carbocation and this is the radical chemistry. So, do not think like there is no photofreeze rearrangements. Just to say you that this type of rearrangements are also there in photochemistry. So, basically we have studied rearrangements, dipie, enone systems, dienone systems and photofreeze rearrangements. That's most of the important rearrangements you cover it. That's all for rearrangements. Okay. So, now what we will do is that we will see another important chemistry, that is your singlet oxygen chemistry. Okay. So, most of the time when you do photochemistry, okay, you have to degasify if you are looking for some other products. Okay. So, you degasify properly in night argon, okay, for at least 10 to 15 minutes, then you do the photochemistry. Otherwise what happens? Oxidation happens, or any other general terms otherwise. Photooxidation, any other thing. So, oxygen basically you have to remove when you do photochemistry, when you are looking for some other important photochemistry. Why? Because once if you do a ketone, if you excite the ketone, what happens? It goes to singlet, then it gets to triplet, right. So, your triplet excited ketone can be easily quenched by your oxygen, because oxygen is in properly triplet state. You have one of the system, very rare system to see in triplet, that is your oxygen. So, you try to excite your ketone and you want that ketone to do some other chemistry like alpha cleavage, but instead of doing that it will react with your oxygen, because oxygen is there, dissolved oxygen is there in the solution. So, immediately the reaction goes off. Type of quenches all your process, because oxygen is in the triplet state. That I ask most of the time this question in the viva, what state oxygen is, okay, in the oxygen in its ground state. This is, I think, one of the main molecule which in ground state exists in triplet. So, it is normally you can write that, yeah, I am not writing any MO theory or something, I am just I can say oxygen, if it is in triplet, I always think them as a radical, okay, for chemistry. But if oxygen is, if you excite this oxygen, it gets to singlet. Okay. Singlet of oxygen normally exists like this. So, this you can call as 1,2 zwitterion. These are very good findings. Okay. Do you think what will be the energy gap between this triplet oxygen and singlet oxygen? Any idea? Whether no, this will tell an idea like whether what process, it is a very hard process or it is a simple process to do it. It's only 22 to 23 kilocalories per mole. Energy is so less. This you know why it is important. When I take the application, you will understand. Okay. So, basically oxygen is ground state is triplet, do not forget that. Once you excite it becomes singlet, that acts more like a 1,2 zwitterion. The energy gap between your singlet and triplet is roughly around 20 to 23 kilocalories per mole. Another important thing which you have to know about singlet oxygen is their lifetime, because any species you want to know their lifetime, that is more important, how much lifetime it is there. So, singlet oxygen lifetime is more solvent dependent, it depends on the solvent lot, okay. But approximately it stays 10 to the power of minus 6 seconds. But if it is in water, it has a lifetime of 2 into 10 to the power of minus 6 second, but if it is in D2O, it increases 20 into 10 to the power of minus 6. If it is in chloroform, it gets 60. If it is in CdCl3, 300 into 10 to the power of minus 6 second. If it is CF3Cl, it is 1000 into 10 to the power of minus 6 second. So, it is highly solvent dependent, but it works around 10 to the power of minus 6 seconds more of the time. So, it is always nice to know the lifetime of your species, so that you can think of many good chemistry like reactions. So, these are some basic aspect of your oxygen. Okay. So, triplet, singlet, energy gap is 22, lifetime is around 10 to the power of minus 6 seconds, some physical aspects. Now, we will see how we can synthesize singlet oxygen, how people synthesize singlet oxygen. Any method synthesis of singlet oxygen? You take your triplet oxygen, yeah, you can use rose Bengal, you can use a commonly triplet sensitizer, okay. You get a singlet sensitizer plus singlet oxygen. Now, basically if you want to look into electron view, you can just look like this. So, you can use rose Bengal, any other which you can think, because you guys are studying bio and organic chemistry a lot. No? Any other guy you can think about which you study? You have studied a lot in biology. Any metalloporphyrins? You can do nice chemistry with metalloporphyrins. People are doing like, you can take alkene, you can take your oxygen, oxygen, you put light and you put a metalloporphyrin, you end up with a Diels-Alder reaction. Okay. This is one common, which is we call as photosensitization, by which you can generate your singlet oxygen using sensitizers. Okay. Nothing big, just you have to take system with an oxygen. Okay, just put your rose Bengal, keep it in the light, you get singlet oxygen, that is all. Nothing more to do on that. Another method which people do is that, heard about chemiluminescence? You call it as thermal decomposition method, but you can call them as chemiluminescence. Heard about luminescence? What is luminescence? Just simply saying, or where you observe this luminescence process in nature? Do you see luminescence process anywhere in the nature? Your fireflies, best example. See, there are many chemistry has been done. Lot of, I think, research groups in Japan work on luminescence. They found out the enzymes which are which can make this light, but still they are struggling to find out how this color transformation takes place. Sometimes it gives yellow, blue, green. That is very important. What type of protein it is trying to do that? That is lot of research are going on that area. But we will take some synthetic way of doing it. Take a proper anthracene. type of system with the phenyl. When you photolyze this, okay, just think you have an oxygen in the system. You get a nice type of yes, from double bond. Oh, sorry, this one, right? Oh, starting material. Okay, okay, sorry, sorry. So this, what happens if you eat this, like 160 degrees Celsius? It gives out your singlet oxygen nicely with your starting material, that you call it as A. It gives you singlet oxygen plus A. And the interesting part is that this also gives light, this reaction. Once you heat, you get your light also out from this. That is how it does luminescence. It basically takes the light, then after some time gives the light back, okay, and that is how people are generating singlet oxygen. But normally, if you want to do in lab, you use your photosensitization method, fine. Okay. So these are the two important two ways by which you synthesize. Now we will see the reactions of singlet oxygen. What are the reactions singlet oxygen does? Any idea what reaction singlet oxygen can do? Huh? It does Diels-Alder 4 plus 2 cycle addition, then it does 2 plus 2. One more important reaction it does. What is this? If I have an alkene like this system, okay, I can write my singlet oxygen like this. Yes, I think I am generated a singlet oxygen. What it does? See, I have an hydrogen. Okay. See, what it does is nicely takes this hydrogen, this can become OH immediately, or like you can, but you can isolate OH. What this reaction is? Yeah, in reaction. This is the first reaction. Okay, you can see lot of in reactions happening with singlet oxygen. Very good example for in reactions. There are many examples. There are a lot of examples where singlet oxygen does in reactions. Okay, so we will take some good examples and see what happens. Again, don't scold me since I am doing with natural product. Now, I just want you to know about this, writing these terpenoid structures. Now, I take this, use my oxygen photolysis in the presence of sensitizer, triplet sensitizer, okay, rose Bengal. What do you expect? what happens? Epoxidation happens. Hydroxylation. Yeah. See, you can think about your nice in reaction, right? You have an allylic hydrogen. And that's what I say. You have a nice allylic hydrogen there. Okay. So you think like you have generated system O minus and plus. So you can come across this. Yeah. Now I will ask you. So you will get a. It can be OOH, which then breaks down to OH. Okay. So whether the stereochemistry will be preserved, like it's a stereospecific reaction, or it is non-stereospecific. It should be stereospecific because I am not talking any radical here, right? Fine. It's a very good example of doing with natural, because people call this type of reactions remote functionalization. It's very hard to functionalize this type of moieties, with an OH. If you think like you can make only OH, it's very hard to do that. You have to make an epoxide, then break it up, then other side is there. Just an ene reaction, and it's very stereospecific also. And there's, if you think about the company point of view, nothing, you have just you are using oxygen, so they will also be happy. Fine. Okay. Now we will take tell me what product you can think of. Can I give a clue? The reaction gives me 3 products. Once I fertilize this in oxygen, the presence of H mu sensitizer, I get a type of 3 products. Shall we write? So, one case you have your O plus, okay, O minus and your O plus. So, you say that you can pick this hydrogen, which is allylic. So, you can take this up, make a double bond, can take this up. This can be in 2 ways. It can be in an upward direction, giving you a OOH like this, fine, or you can write it like this also. You can write like this also, fine. That's another product you can look for. What is the another product you can think of? Yeah, I can pick an hydrogen from this. This is also allylic, right? See, I am just writing OOH, just you should remember that it can be OH immediately, because normally when people give you this and give you OH, you should think that it's a type of in reaction. Okay, don't look for OOH all the time. This OH can easily break and become OH. So, your yin reaction should come. I need a double bond. Yeah. So, you get 3 products nicely. Okay. So, this is one type of reaction of first reaction of your singlet oxygen yin reaction. So, singlet oxygen does nice yin reaction. Okay. So, we will see the next type of reaction which is singlet oxygen does, that you call as Diels-Alder reaction, or you can call as 4+2 cycloaddition. I can generate a singlet oxygen. Okay. So, I can do my nice Diels-Alder. You call this as endo peroxide. Okay, if you take this product, because just want to check about the what do you get? Now, why I am writing this, just to check up the stereochemistry, because this is a Diels-Alder, then it reaction is type of very stereospecific. So, I am just worried how you write that method, that is what this example is all about. Just to show that yes, this reaction is very stereospecific, highly stereospecific. Fine. Now, we will take another very good example which you can see how Diels-Alder works. Finish. Okay. So, it is better that you do lot of problems, that will help you out. Write the product. So, you wrote a right. So, you were thinking about this one, yeah. That's the interesting part. You can see what happens. This guy started flipping. Okay, it can around cis and trans and all these things happen. So, it does not allow you to come like this to make a OO bond. There are examples which I will show, but this is easy. This cannot do anything. Okay, so oxygen can sit nicely inside and make an Diels-Alder there. Fine. No, if you are thinking like this, it is not wrong, it is right. Okay, do not think that you are completely wrong. You are thinking rightly if you are doing that also. Okay. The next is that you have to see why it is wrong. Okay. This is interesting one, because this has been recently done, this work. We will check how many has written the right one for this. I want the product for this. Looks to be a very simple molecule, okay, nothing big there. Can you give me the final product? You have written the product. Just simply a Diels-Alder, that is all. Written. Benzene ring, benzene ring, any other? Yeah, that is not wrong. See, if you basically are stopped from there, it is not wrong that you get a benzene plus you get one more cycloride that you are not thought about. See what happens, your first step is what? That molecule thinks nice than us. Eh? Now again you have a system which is ready for Diels-Alder. So that again can give you this. So basically you should not stop that. These are good examples you should learn about doing things, Diels-Alder reactions. I will see Diels-Alder reaction of oxygen in heterocyclic systems, very famous. You can it is used for many synthetic utilities. Like, see, there are few reactions which you can, for example, I have a furan. Okay, I want to do functionalization here. You can do by using, this is normally done in even synthetic labs. This you can do because these reactions are normally done easy. Even if you are doing in your project, you can think of doing that. You can come to my lab. There are, I think previous year also some project student did this reaction. He came to my lab and did this reaction. Very simple in one sense. Like, you get this, right? Once you do the Diels-Alder. If you slightly warm this, once you take it out and slightly warm it, immediately you can get these products. Then you can break this epoxy to give you a OH. You can get these two products. Basically, you can get opening up on both the sides and making epoxide, or you can get a ketone with an epoxide. While warming, if you warm it using methanol, slightly if you warm with methanol, then you end up with the ketone. You get this product. While if you want to warm it with methanol slowly. So, you can functionalize your ketones. There are some more reactions. If you want to warm using little bit triphenyl phosphine, then you get nice dialdehydes. Can get, you take, because furans are cheaper, take furan, do a singlet oxygen, put lightly warm it with triphenyl phosphine, you get dialdehydes. Then you can do Wittig or whatever you want, you can extend the chain. Even you have reactions with thiophene, you can do with thiophene also, not only with furans, you can do with thiophene. You can same way you can get sulfur and the same products. These are like important reactions of your singlet oxygen in Diels-Alder. Fine. Okay. Now we will see the third one that you know by yourself, like that is your 1,2 cycle addition, right. This is just for previously, oh sorry, not 1 2, 2 plus 2. You have seen 4 plus 2 cycle addition, which is your Diels-Alder. Singlet oxygen does also 2 plus 2. You get 1 2 dioxytanes. Molecules. Right. So you have studied basically 3 reactions in singlet oxygen. One you have studied in reaction, another Diels-Alder, and another is 2 plus 2 cycle addition. Fine. Okay. Now I will give you one simple example and ask you how to see what you can take this molecule, like tell me what products you can think of. I am taking an type of diene system, I am using oxygen photolysis in presence of sensitizer. What you can think in this reaction? What products this can give you? All three, very good. You get all 3 of your reactions here. You get in reaction, you can think about 4 plus 2, you can think about your 2 plus 2. Very good example for this, very good. So, you get all 3. Can you write the product? This you can get from your in, right. So, I can take this hydrogen from this product, I am right. Yes, I can get this product, then I can think about nice 4 plus 2 cycle addition. I can get my 2 plus 2 cycle addition. These are very good examples to understand about your chemistry. Done. Okay. So, I will take another same type of molecule. Say I am just giving a molecule Okay, I have a molecule like this. This is the other way around. Okay, one I have a methyl hydrogen, another I have hydrogen methyl. It's just a cis and trans of it. I am doing oxygen photolysis sensitizer. Same condition I am following for this. Same molecule I am taking, I am following the same condition. So, in which what will happen? In which case you are thinking about ene reaction, and in which case you are thinking about 4+2 cycloaddition. See, this gives among these two molecules, one give only ene reaction, another molecule gives only 4+2 cycloaddition. Yeah, anyone can tell. You say? Okay, we call it as A and B so that we don't get confused. So, you say A as in reaction. Why? Any idea? See, you just see here, okay, this hydrogen is there, right? The interaction of this hydrogen and methyl, this interaction of this hydrogen and hydrogen, which is more? This is greater, okay. So, what happens? It does not want to stay there, it just flip across, it goes down and flip across, okay. So, you get a structures, I will just say similar to a structure. Facing this direction, okay, it moves away, fine, with the hydrogen and methyl this direction, which is completely away, it goes flips around, okay. In this case, it does not happen. Now, this is not fissile for doing a 4+2, okay, it can do only in reaction, okay. In this case, you can think about nice 4+2 cycloaddition, because it will be on that side. So, oxygen can come and bind and get you a nice 4+2 cycloaddition. So, in this case, if I write the product, I can get this product, right. Can get this product in fine. So, more about the like substrate also has more control in which the reaction wants, ene or 2+2 or 4+2. In some cases you see all these 3 reactions happening, in some case 2. For example, if you take this type of systems, what you can think in this case? Oxygen photolysis sensitizer. O tertiary butyl, okay, TBU, tertiary butyl, TBU. What do you expect in this case? You expect 4 plus 2. If that means I have to write the structure like right. You expect this. Why cannot I write the structure like this, which will be much more easier for me? Why cannot I think about writing like this? 2 plus 2. You can think of writing like this. Anything wrong in this? Whether this undergoes, so it undergoes both 4 plus 2 and 2 plus 2. That's the only thing in these reactions. Okay, you can see 4 plus 2 as plus 2 plus 2. See, that's why I say there are many good examples where you can see all 3 reactions happening. In some cases 2 happening. In some cases it is very selective for 1 reactions. Okay, but you should remember that if you have a singlet oxygen, you think about ene, Diels adder and 2 plus 2. Fine. Okay. Now, the last reaction of your singlet oxygen, which is now well explored and people are using for a lot of big applications. Sorry. Okay. The last application which we are talking about is PET chemistry. Okay, if see this is this has been this application, this particular reaction application has been taken now. It's in basically in hospitals. Okay, they have been directly taken into now people are using it. Singlet oxygen plus if you have a donor, you generate singlet oxygen. Okay, you have a donor. So what happens? A donor can give an electron. That's your PET chemistry, right? So you get singlet plus D dot plus, right? Okay, I just take this out, this one O2. What do you call this? Very famous compound. Yeah, this is superoxide, highly reactive species. Just you take singlet oxygen plus your donor, you get your superoxide. Okay, now I want to ask is where this chemistry is used. I have studied this chemistry, a lot of application it has. This particular superoxide. Anyone?
Evolution of oxygen gas.
More than that. What is photodynamic therapy? Heard about this? Photodynamic therapy. You guys know about like how to kill cancer cells, right? Cancer therapy. How they normally do? Normally in hospital, if you have a cancer, what they do? Radiation therapy. Radiation, because what is the big drawback of radiation? Yeah, it kills your good cells also, and as a result of that, you might end up with some other problems. So, but in this photodynamic therapy is so nice that what happens is that, for example, you have a cancer here, okay, and you want to kill only that area. So, what you do, you take a dye, inject that dye into that particular cell, okay, and then cover your whole area by black cloth or whatever, okay, and make a small hole there. where you have the cancer. Okay. Then shine light. So, what happens? Your dye gets excited. No. This excited dye will be then interacting with your oxygen, because that is there in your body, interacts with an oxygen to give you singlet oxygen. Okay. This singlet oxygen then react with your DNA or protein, whatever comes into its path, and this DNA acts more like a donor, okay, generating you O2 minus dot plus DNA plus dot. So, and this superoxide anion will just start killing the cells. It kills all the unwanted cells in that particular area, because your dye is not other plant, your dye is only on that area. No? So, it just kills on the unwanted cells. Okay. That is called photodynamic therapy, that is now started using in field. But the only drawback of this system is that it is more in the like skin cancer, like peripheral region. If you have something inside like kidney or liver, wherever it is, then this does not work, that is the only drawback. But if you have on the peripheral regions, this is the best thing. You just because it is more target oriented, just you take the dye, inject on the particular area, and just then that is all, just shine your light, that is enough. Then you get your unwanted cells killed. And many schools, okay, many schools have been started, centers only on photodynamic therapies. Okay. Bowling Green State Universities, there are University of Leeds and many other universities, they have started only center where they are doing only photodynamic therapies. Okay. So, that is 4 important reactions of your singlet oxygen. So, the chemistry of singlet oxygen is that is. So, we have seen what singlet oxygen, their synthesis, and we have seen their 4 important reactions. Fine, that is good. So, we are ending our class with this.